We have work going on within our USP committee that addresses obviously the measurement of contamination in products, which maybe has been our historical focus. But over the last several years, we've become increasingly aware of the fact that we need to focus more of our energies on helping our stakeholders develop systems for the avoidance of contamination and to be better aware of the conditions within their own facilities or within their own manufacturing processes that might favor microbial contamination. So I think one of our current points of emphasis is on helping firms develop better means for facility hygiene, process sanitization, that will enable them to avoid the pitfalls of microbial contamination in products or in bioburden for sterile manufacturing. So there's a great deal of emphasis on that. Our committee works in the area of microbiological contamination control for both sterile and non-sterile products, and we have major initiatives underway in both areas. So I think there's going to be something for the microbiologist at this meeting, whether their primary responsibility are sterile products or whether they are non-sterile products. I think they'll find things here of interest to them. We're interested in making methods more available within the USP that allow a more rapid assessment of contamination or contamination favoring conditions within processes, equipment, ingredients such as water, excipients, or active pharmaceutical ingredients. So those are certainly things that we want to be able to discuss with our stakeholders and also to take advantage of the expertise and experience that they've accrued over the years in working in this area. So we see our conference as an opportunity to bring that together and to have very frank and open discussions with participants regarding the control of contamination and what are some of what input they can give us regarding the problems they may face on a day-to-day basis. In a very general sense, having rapid methods available for the assessment of contamination enables a microbiologist to make more timely decisions regarding, in some cases, even the continuation of a production process. In fact, the advantages of rapid methods are almost limitless. We have new products entering the market that are cytotherapy based, regenerative medicine products that are made specifically maybe made specifically for an individual and which are utilized over a very short period of time. And our current 14-day sterility test, for example, would be inapplicable because the test results would not be available until long after the product is utilized. That would be true of other biologics as well. It's certainly true of radiopharmaceuticals, positive emission tomography products. So there are a number of products, and I think more products that are likely to be introduced in coming years for which very lengthy microbial test time would be not just disadvantageous, but actually impossible to tolerate. We see that our ability to communicate with our stakeholders is vital, and it provides two opportunities. One is for us to share with the participants our current thinking and what progress we've made since our last stakeholder intervention or our last stakeholder discussions regarding the development of new chapters, new informational works to enable them to better implement technologies. And we also then can get input from them as to whether or not they think we're on the right track.